All right, so this week's lab, the question that you're asking is sodium chloride a buffer, right? That's our scientific question that we're dealing with this week. Now, before coming up with a hypothesis, remember, you need to come up with a hypothesis, a hypothetical answer to this scientific question, but you do so before you do any experiments. Okay, so you normally won't have any data before you see this. And so before coming up with a hypothesis, what you want to do is a little academic research. So let's look into what buffers are. Well, before we can look into what buffers are, we have to understand the concept that the concentration of hydrogen ions in solution is an important factor to know. As you will learn in lecture, it is what sort of determines whether a protein is in its native conformation or whether it's been denatured. Okay, so that's affected by the hydrogen ion concentration. And so we're constantly talking about it in biology and biochemistry. The problem is to say something like, oh, the hydrogen ion concentration of this solution is 1 times 10 to the minus 3 molar. Or the hydrogen ion concentration of this solution is 1 times 10 to the minus 9th molar. That's a mouthful. And we're talking about it all the time, and we don't want to have to constantly say all of that. So instead, what scientists did is they said, all right, let's just make the pH equal to the negative log of the hydrogen ion concentration. Then, instead of saying the hydrogen ion concentration is 1 times 10 to the negative third, all we need to say is that solution is pH 3. They mean 100% identical things. It's just a different way of saying it. Okay? And this here, the hydrogen ion concentration 1 times 10 to the ninth, means it's just pH 9. But be careful, because this negative log relationship here, if we have something that is a higher hydrogen ion concentration, 1 times 10 to the minus third, it's going to, because of that negative log, it's going to have a lower pH, right? So it's just kind of weird. And solutions that have a lower hydrogen ion concentration are going to have a higher pH. Okay? Doesn't mean they're not talking about the same thing. They totally are still talking about the same thing. It's just the relationship is a little inverted because of that negative log relationship. All right, so what that means is solutions of lower pH have higher hydrogen ion concentration than solutions of higher pH. But both of them are simply measurements of hydrogen ion concentration in solution. Now, buffers are solutions that minimize changes in pH, all right? Like I said, it's really important for the structure of proteins to have to be at a very specific pH. If you go too high or if you go too low, outside of the optimal pH of that protein, you're going to denature it. And so in the body, there are buffers that minimize changes in pH so that these proteins will not denature. And one of those buffers is carbonic acid. And like all buffers, carbonic acid is a weak acid. Okay, so first of all, an acid is a molecule that when you put it into solution, it increases the hydrogen ion concentration of that solution. It deprotonates. Now, the fact that it's a weak acid means it doesn't fully deprotonate. So when you look in a solution that has some carbonic acid in it, you are going to see some that are deprotonated. So here's the H plus that came off, and here is the conjugate base, right, the HCO3 negative. And then there will be a portion that remain protonated. And this is why weak acids are good buffers, because in the same solution, we have a way to react if an excess of protons have been added or if protons have been taken away. For example, if protons are taken out of solution, then what happens is some of the acid that didn't dissociate, that didn't deprotonate, now deprotonates and resupplies those missing protons. Or if we added an acid to this solution and we've increased the hydrogen ion concentration, this weak acid, this buffer, can suck up some of those those protons, those excess protons, by binding to the proton acceptor and making more of the protonated state. Okay, so weak acids can react in either direction, sucking up excess protons or donating protons when some have been taken away. And in doing that, they minimize changes in pH. So, again, our question is sodium chloride a buffer? Take a look at sodium chloride. Take a look at what happens when you put it into an aqueous solution. Does it look like a weak acid? Okay, can it do everything that we've just been talking about? So regardless of what your hypothesis is, you're going to be doing three things. Okay? You're going to be adding acid and base to your non-buffer control. That's water. And then you're going to see, well, what's the change in pH when I do that, when I add the acid, when I add the base? Then you're going to do your other control. Now, your other control is a known buffer. It's going to be sodium acetate. And you're going to add acid and base to it, and you're going to see, well, how does that affect the pH? And then you're going to do your experimental. You're going to add acid and base to that sodium chloride. You're going to measure the change in pH, and you're going to compare that to your non-buffer control and your buffer control. And you're going to see which one of those two it more behaves like, that it behaves more like. Okay, so this will be the example of the data that you would get from this. So for example, okay, before you add any acid, here's the current pH of each of the three items, right? Our non-buffer, our buffer control, and our sodium chloride, our experimental. And then this is what happens after you added 250 microliters of a strong acid. The pH has gone down, the pH has gone down, the pH has gone down. The question is, let's analyze that data. How much did the pH change, right? Because that's really the measurement we want to focus on. Now, when you analyze your data, you're going to do it in table 2.3. And let's take a look here. We're going to have some sample data that we started off at 7.4. We added the acid, and we ended up at 6.2. So here's how you analyze the data. Did the pH, what was the pH? Did the pH increase or decrease? Well, yeah, I mean, 6.4 is less than 7.4, so the pH decreased. What was the total change? Well, subtract the two, put the number there. Do not use a minus sign or a plus sign at this step, okay? Keep it as just the absolute value. Did the hydrogen ion concentration increase or decrease? Well, remember, if the pH decreases, that means the hydrogen ion concentration increased, right? Because there's that sort of inverted relationship between there. Now, if you want to figure out the fold change in hydrogen ion concentration, every 1 unit of pH change is a 10-fold difference. So if it was just 1 pH change, then it would be 10-fold difference. In this case, it was 1.2 pH change. The fold difference would be 10 to the power of 1.2. And then down here, all we want you to do is take 10, put it to the power of 1.2, and that'll give you a just a straight up number for the fold change instead of having it as represented as a power. So that is the data then that you are going to analyze. And then you're going to, right, you're going to come up with a discussion. You're going to describe what happened, and then you'll come up with a conclusion on whether or not you're going to support your hypothesis, or you have to reject your hypothesis. Now, all of that is in the Google Docs assignment that you and your lab mates are going to be working on together. I want nice, full sentences when they are asked for. Be brief, but make sure you are thorough.